this demo, I show how I use Claude Science to turn an AD epigenomics question into biological insights, candidate targets, and testable hypotheses. I first configure a custom single-cell lead specialist to run the analysis. I gave it clear responsibilities, reproducible standards, and revision rules, so every step is traceable. I can annotate results, request missing analysis, or redirect interpretation while keeping a record. Everything is driven by a structured project file defining the questions, data, and standards. Then I just tell it, follow the file, and it runs the analysis and organizes outputs cleanly. Claude Science generated a fully reproducible workflow and a report that reads like a concise paper. It built an atlas across 2 histone marks that I can reuse in ongoing work. And it identified differential peaks. When those signals were subtle, it stepped up to the gene level, revealing meaningful pathways, and it further characterized enhancer gene links and glial subtype changes. As part of my review, I annotated the report and asked for revisions. For example, the initial discussion leaned on cerebellar microglia immune programs. I asked for a deeper hippocampal interpretation because it is more vulnerable. The revised discussion shifted the biological focus, and one finding I found especially compelling was a hippocampal astrocyte state switch, with homeostatic programs reduced and reactive inflammatory programs increased. Claude Science summarized the evidence and connected to prior literature. With analysis complete, I asked Claude Science for 2 more things. First, target prioritization. It called a key issue. Initial gene expression was based on aggregated chromatin signals, so it was revised to incorporate RNA expression. It returned 80 risk genes supported by both enhancers and RNA in each cell type. For example, SORL1 in AD microglia show reduced expression together with enhancer loss, suggesting impaired lysosomal function and A-beta clearance capacity. This is what we can test first. Second, I asked for a grand outline. I wanted to know what drives the hippocampal astrocyte state switch. Then I got 3 connected but independent aims: where it occurs, how pathology drives it, and why it matters. For each aim, it pairs supporting pilot evidence with proposed experiments like spatial transcriptomics and perturbations. So this is how Claude Science helped me move from question to findings to targets to a proposal.